I would like to do today is I'd like to share with you different paradigms for improving children's health. And I'm really going to focus on obesity prevention. And we recognize how incredibly important that is when we look at diabetes prevention. So I'd like to start today with a story. It was never good when the red phone rang. It meant that something unexpected was about to roll into the emergency room. On this hot summer day, it was a 2-year-old, the victim of a drive-by stray bullet that lodged into his brain while he slept in his mother's arms. And I happened to be the first responder as a second-year resident at Children's Hospital Los Angeles. This was Los Angeles in the 90s, and we did what we could do. We stabilized the patient. We stemmed the flow of blood. We stabilized the patient, but we didn't solve the problem. And for me, that was my personal aha moment. We all have that when we choose how we ask questions and what problems we're hoping to solve together. So that made me ask questions about what does it mean to solve the root problem? What do you need to understand so you can understand the root problem? And so today, what I would like to do is I'm going to describe and differentiate key components of a complex versus a complicated system. And you'll hear about how I applied complexity science when it comes to my research. I'd like to explain the principles of life course. I suspect many of you already know this. Just if you could raise your hand or give me a thumbs up if that's a familiar concept to you. Yeah, thank you. So then what I've done in with my team is we have taken the combination of a complex systems thinking approach plus life course so that we could then understand what are the drivers of childhood obesity and all of the common chronic conditions, including diabetes, that result. So we know as we are solving problems that we are left-brained and right-brained. And often the left brain about being sort of linear and rational and objective is really different than what we see when we are trying to zoom out and solve problems in new ways. So a traditional way for us to solve problems, it's based on Newtonian paradigms. There's a linear cause and effect. It's a very specific mathematical model. We've all used this in solving problems. But in complexity science, that means we're looking at systems where unexpected patterns emerge as a result of many individual parts interacting. So the interacting parts follow a set of rules that result in emergent properties. So you need to ask different questions. You want to understand how does each individual part behave in the system? And then how do the parts of the system act together to affect the function of the whole system over time? So the best example of that is when you see a flock of birds in the sky. So familiar scene, the sun is setting, you look up and you see this flock of birds and they're moving in these unexpected patterns. And you have to ask a lot of questions. Who's the leader? How is it that we know where they're going? Why don't they bump into each other? And the way we understand this is by recognizing that it's a complex system. So I want to compare a complicated system to a complex system because sometimes we use these words as if they're synonymous, and they really aren't. So a complicated system, I want you to think about a Swiss watch. So here you're going to be thinking about homogeneous, identical structural elements. Every Swiss watch looks the same. You look at linear relationships. So these are relationships with constant proportions, and that's why it keeps time precisely. It's deterministic. Same result always occurs for a given set of circumstances, so it's predictable. Static, nothing is changing over time. Independent, subsystems aren't influenced or controlled by other parts of the systems. The gears fit together. And there's no feedback. There's no there's no feedback loop to change what's happening in this system. It will be the same reliable system every time. Now, many of us know that this doesn't apply to people or living systems. Many living systems are complex. So when we look at complex systems, I want you to think about a beehive. So in a beehive, you see that it's heterogeneous. There are a large number of structural variations. No hive actually looks exactly the same. It's nonlinear. One member in the colony doesn't always produce the same and proportional effect as another. It's stochastic because there are always elements of randomness that leads to degrees of uncertainty about the outcome. There are dynamic changes over time, and the past has an impact on the future. If you had a colony that wasn't thriving, it will affect what comes next. It's interdependent. Subsystems are interconnected. They're interwoven. They're not just interacting. And there's a feedback loop that will determine how much honey is produced or not produced over time. That's a complex system. And I want you to remember these components because I'm going to be calling them out as a framework. So, large number of variations, non-linear effects, randomness, dynamic, feedback loop. So, what's interesting to me is that we recognize when we are looking at patterns, whether there are patterns that are emerging from a flock of birds in the sky, or we look beyond that and see the pinwheel galaxy, the reason why things function in a certain way is it's usually the most efficient way to transmit information. And this is why we often see similar patterns, whether they are as small as your fingerprint or as large as the pinwheel galaxy. And of course, everybody recognizes this as the Fibonacci sequence. So that mathematical property actually is translated because it's a system that's designed to have an efficient flow of information. So we in my team, we've applied that way of thinking whether we're looking at epigenetics and the signatures above the genome, or we're looking at a broader zoom out of social networks and how information is exchanged between people and communities, and we combine them together. So I actually think about when I am looking at complex systems and how they meet life course, which we'll talk about in just a minute, it reminds me about Daniel Kahneman's book, Thinking Fast and Thinking Slow, recognizing that we have these two different ways, two different systems. I'm sure it's more than that. And they're operating at the same time. Either you have system one thinking, which is fast and automatic, or you have that slow and deliberate thinking. And we have to determine which approach and which system we're using when we're solving problems. So, first thing that we're building on here is that we're looking at complex systems. So we're looking at each of those elements of a dynamic, heterogeneous, nonlinear system that has a feedback loop. We are then adding to that a life course perspective. And I'm in pediatrics for a reason. I really believe that in children, you have an opportunity because of developmental plasticity to change trajectories away from disease and towards health. But we have to understand all of the components of the complex system to do it well. So just for those of you who are less familiar with life course, this is work around type 2 diabetes. We recognized on the x-axis that the younger you are, the more developmental plasticity you have. And the older you are, by the way, look, you still have developmental plasticity as you get older. Don't fret. But you can see that your response to new challenges will be sort of hardwired over time. And so we're trying to understand what is the right timing, what is the right dose, what is the right duration for interventions to be effective. So if we're able to understand, as we now describe, that we recognize obesity as it's a condition of chronic inflammation. Well, if we can step it back and understand what sets those systems up in mothers and infants, well, then we could potentially change that outcome. So we're not in a pro-inflammatory state. And then that doesn't lead to chronic conditions like type 2 diabetes. It doesn't mean we shouldn't intervene in childhood and adolescence. And my team, we tend to focus on preschool periods of life. It just means that the intervention, the timing will affect the dose and duration, and the outcome will have a different delta. So if you're intervening sooner, you can have a much higher delta than if you're intervening later. So that's the basic concept of life course. Timing, dose, and duration matters. So often when we think about life course, we only think about timing, but that by itself doesn't explain what you see. So how much of an exposure over what period of that time? So you want to capture all those elements. So a life course informed model applies complexity science and allows us to now introduce new ways of thinking about diseases that develop and new approaches for treatment and prevention. So as I move on, I'm going to combine these two frameworks now. So how do we look at complex systems and life course at the same time as we're understanding what causes are, what potential interventions are, and how we can get to improved health outcomes in childhood and into adulthood. So a lot of people will say, you know, I like this theoretically, but how do you actually analyze such complex models and frameworks? You can't distill them down to the same sorts of analyses that many of us are sort of trained up in. So an example here is when you're looking at simple correlations. This was such an important discovery in 2008. This is when we realized, fat mass seems to be related to the FTO gene. And everyone was now thinking, it's all about the gene, it's the FTO gene. But you can see here, we basically said, what's one thing that's affecting fat mass? And this is one thing, but it's not the only thing. And depending on your context, it might have a different phenotype that emerges. Because of that, my team uses the Hong and Glass model, which is a systems-oriented multi-level framework. So we look at this micro level, and the micro level includes things like the HPA axis, mood, appetite, metabolism, and genes. I'll focus today on epigenetics. Then there's a meso level. The meso level is what makes me popular at parties because everybody knows how they can solve obesity. You should just eat less and you should move more. And then I remind them that they probably know people who've tried that and it's worked, and they've probably known people who've tried it and it hasn't worked because we're complex systems. So on the meso level, we talk about behaviors, nutrition, physical activity, sleep, and we look at this with children in the context of their parents. And then at the macro level, that includes things like cultural norms, built environments, area deprivation, local food environments, psychosocial hazards, and commercial messaging, just to name a few. And I'll talk about built environment as well as what I consider to be in between the meso level and the macro level, and that's social networks and how we influence each other. When I say social networks, I don't mean social media. I mean people that you're looking at them in the eyes and you are asking them to do something for you. Maybe you're watering my plants when I'm out of town, and maybe I'm letting your dog out. So it's about the ties that we make between people. They can be weak or strong. So I'm using this model And I'm going to focus on epigenetics at the micro level. We'll talk about energy input and energy expenditures and social networks at the meso level, and then built environment. Now, then you have to actually analyze all this information. And you have to recognize that you have feedback loops and you have to look across time because depending on when you're exposed to any of the things we're about to discuss, that timing, dose, and duration really matters in terms of outcomes and how we can improve health. So I'm going to start at the micro level. And we're quite familiar that genetics doesn't change very quickly, but epigenetics, which is changes in gene expression via post-translational and post-transcriptional modifications, well, that's something that appears to be plastic. Now, we're not exactly sure for how long, and we're not sure at what point do we actually have a set point that has been established. But we do know there's a period of plasticity where the marks above the genome, whether we talk about methylation or histone modification or the many other things that we can talk about now because of how this field has grown, that these are the pencil marks that can be erased over time until they become pen marks. So when I explain epigenetics to people who don't understand it, I usually say you can't change your genes, but you can change the way your genes behave. So here's what my team did in regards to trying to understand epigenetics in children that were at risk for obesity, but not yet obese. So we took preschool child saliva, and the reason why I collected saliva as the tissue and not blood is that I was in community settings, and salivary tissue was much more accessible and less scary to the participants. So we took child saliva. I needed to be able to annotate it and understand what it meant. And then I asked the question, well, if I look at boys and girls and we put them in gender and aged matched pairs, and the only thing that's different about them at that moment in time when they spit into the cup is that their maternal BMI is different. So either the maternal BMI has a visceral adiposity of greater than 100 centimeters, or it's less than that. So we look to see, do you have obesity, maternal obesity, or do you have a lean, normal BMI as a mother? Then what we did was we said, okay, these children, none of these children are obese, but they are at risk for obesity because we enrolled them in the study with a BMI greater than or equal to the 50th percentile, but less than the 95th. And they all came from lower SES environments and were on some sort of supplemental program. Both of those things are correlated with later obesity. And what we saw, even though children were not yet obese, is that 119 CPG sites were significantly associated with maternal BMI. And of the 119 CPG sites, 41 were found within a transcription start site or another regulatory region outside of the gene body. So many of these sites were associated with genes that had biologic plausibility in relationship to obesity. And I'm just going to give you a little tiny taste of what those were. So we saw that these are just exemplar findings from our data that reflected statistically significant effects of maternal BMI. And we saw melanin concentrating hormone receptors differences. So that's integral to plasma membrane protein. And many of you know this because it's involved in neuronal regulation of food consumption. We also saw that the site associated with PCSK9 was different as well, and that regulates the amount of cholesterol in the bloodstream. We also saw changes in chemokine receptors and changes in the NLR family. And this specifically, this pyrine domain that contains 6 is part of an inflammasome complex. So it's involved in your gut microbiota's sensing capabilities for inflammation. So that was really intriguing to us because now you begin to say, I wonder how these things are affected when they're affected. And if we intervene, at a period of plasticity, could we reverse those epigenetics, those marks? What would that look like? So just want to check in with you all to make sure. Thank you. Can you all hear me? Just give me a thumbs up. And I am going to ask if you all wouldn't mind if you see something in the chat, feel free to pause and let me know if there's an opportunity for a conversation. Otherwise, I'll wait to the end. So now we've got a lot of interesting biologic plausibility, and we were able to ask the question, well, does this information, so this is before any child, it was at baseline, became obese. This was an 8-year study. Every child was in the study in a rolling 3-year period of time. And we were able to say 3 years later who emerged into obesity. So we went back to our saliva and we said, could we have predicted who emerged into obesity, regardless of what they ate, their physical activity, and their sleep behaviors, based on these methylation marks? And we saw, and here this is, I matched it to the pilot, which is why these data are exciting, but I would not say they're confirmed. It gives us directions for what to do next. What we saw was we looked at those children that became obese, and 30 of them became obese in this original sample that I used, versus non-obese at 36 months and 48 there. And we had to make sure that we looked at age, because age and gender have an effect on epigenetics. So you can see the difference here in their BMI and BMI Z-score here. And we had a preponderance of families that describe themselves as Hispanic Mexican. We also did descendancy with our assessment because we had Hispanic non-Mexican as well. In fact, we had many different descendancies in our children that identified as Hispanic, which is also an important element to look at. So what we saw once we looked to see could we have predicted with some CPG baseline methylation who would become obese 3 years later, regardless of nutrition, physical activity, and sleep? And the answer was yes. In fact, it looked like NRF1, which is an innate immune response governing adipocyte inflammation, cytokine expression, and insulin resistance. You could see that there's a threefold odds that if this is where we saw the epigenetic mark, that that child 3 years later would emerge into obesity. And that allows us to ask really different questions, because now I can look when children are much younger to understand epigenetics around this site to determine how we might intervene to affect how we consider our cytokines and adipocyte inflammation. So it changes the way we think about things. The other thing that was really interesting to me is we looked at top canonical signaling pathways. And you can see that these were the top signaling pathways. And the ones that really stood out to me was all of them actually are interesting, but circadian rhythm really stood out to me as something that really hadn't at that time been studied very much. We know that 40% of our genes respond to circadian rhythm making. It's a key driver of physiologic functioning. And this really made our team ask additional questions about circadian rhythm. And remember, when we talk about circadian rhythm, it's not the way that I learned about it when I was in medical school, where we thought about the master clock and the pineal gland. Well, now we have this deep understanding that every cell has circadian rhythm. Certainly, every organ has its own circadian rhythm. And the question is, if we're asynchronous in our organ system, does that actually create more inflammation and then create this pathway towards all things associated with chronic inflammation, like type 2 diabetes and other things? So that changes the way you think about what an intervention is. It's not only what you're eating, it's when you're eating. and when you're sleeping. So we were seeing in this work with a pretty small sample size for that epigenetics, although I've got all my saliva banked for 610 children, and that is a study that will be in the future waiting to happen, is we're seeing differences in circadian rhythm even before obesity emerges. Now, what I'm going to do next is I'm making the sandwich because first we were talking about epigenetics at the micro level and what might be shifting and changing. Then I'm going to take us up to the macro level and I'm going to talk about built environment. The reason why I'm doing that next is everybody thinks about behavior, but I want you to think about how all of these levels are interacting and moving over time with a complex systems way of thinking. So the Growing Right On to Wellness study was 610 parent preschool child pairs. They entered the study, the children, when they were 3 to 5 and exited when they were 6 to 9. It's an incredibly deeply phenotyped cohort. We've got an incredible amount of data. I had everybody wear accelerometers, parents and children. And the kids didn't want to take them off because we turned them into superhero belts or princess belts. And so the wear time is spectacular. And we were able to understand a lot about behavior. That's just one example. We did 3-day diet recall as well for our kids over time, and it was collected over this 4-year period of time at baseline 1, 2, and 3 years. So everybody goes right to nutrition and physical activity, but often we forget that our behavior is very much shaped by our environment. So this study was done when I was in Nashville, and I saw Rosette's on, who Rosette and I had a chance to work together when we were at Vanderbilt. And your built environment is really important because it affects how we interact with our environment. So neighborhoods and communities made infrastructure and environmental context that affects our interaction with the environment. That's what I mean by built environment. Might include green space, sidewalks, traffic density and speed, crime as an example. And what we wanted to know was, did things change over time? What I'm showing you here is actually, these are the baseline years of the GROW trial. And we wanted to know, this is geographic information systems. Why were parents not sending their children outside to play? And that was a natural experiment that I walked into. Would people use that space to be physically active? And would families use it? So the answer was that whether you were in North Nashville, and I'm calling this North Nashville if it's north of the Cumberland River, and South Nashville if it's south, both in North and South Nashville, parents equally were not sending their children outside to play. They weren't using their built environment for physical activity. So the question was why? And one of the top things that we think about is, well, it's got to be crime, right? So warmer colors mean higher crime. What you see with the stars, we partnered with Parks and Recreation. And so you can see that we actually recruited people from 5 miles within their parks and recreation space, with the idea being behavioral to environmental synergy to change behavior patterns like physical activity. So you can see this is not unusual. Along an urban corridor, you have higher crime rate, but you can see that North Nashville had higher crime rates than South Nashville, but parents equally were not sending their child outside to play. So we decided, instead of going to the literature, to go to our patients. And we said, tell us a little bit about what prevents you from sending your child outside to play. And we asked our families in North Nashville and South Nashville. And in North Nashville, we were right, it was crime. But in South Nashville, it was something we hadn't considered, and that was stray dogs. So what you're seeing here, the difference between this green and this red, is here you have 40 stray dogs, and here you have 4,000 stray dogs. And why is that important? Well, that's important because we already had policies in Nashville. They just needed to be enforced. So I was able to take these data and present it to our mayor at that time, Carl Dean, to say, we've got this amazing resource in our city. More people, more families would use it for its intended purposes of being healthy and well if we enforce the stray dog policy. So now I'm going to be moving down. I gave you the micro level of the epigenetics, the macro level of a built environment. I'm moving into the sandwich part, the meso level. And here I want to talk about what it is when you're looking at social networks. This is what a social network looks like. It's sort of a typology, and you can turn it around three-dimensionally too to see how these lines work. But if you're a larger yellow bubble, that means that the BMI is higher. And if you are that green bubble, it means that you're lean. And then you can see the strength of the ties and the types of the ties here. This was work by Christakis and Fowler that really showed us that it looked like obesity seemed to be contagious when you looked at social networks. And so specifically, and I want to explain a little bit about social networks because we use it intentionally in our behavioral interventions, and I'll tell you why in a minute. So I want you to understand what it is. So a network is different than a group. It implies a specific pattern of connections between people. The typology is the shape. I know we've heard of 6 degrees of separation, but in social networks, it's 3 degrees of influence, where you look at behaviors and ideas and norms that flow across ties. So friend of a friend might affect what I'm trying. I recently started increasing more protein into my diet at this age of my life. And I decided to do it because a friend of a friend told me what they had experienced. That's 3 degrees of influence. Transitivity is multiple connections between nodes and tendency of a friend to become a friend. And then homophily, which is the tendency of people to associate with people who resemble us. And what's so interesting about homophily is that it's completely subconscious. If a friend becomes obese, your chances of becoming obese increases 57%. So among mutual friends, your chances are much higher. Same genders have more influence, and there's a limited effective distance. Whereas if a sibling becomes obese, your chances of becoming obese increase 40%. And then if a spouse becomes obese, it's 37%. So you can see that your friends really make a difference. Now, when we look about typology and diffusion of obesity prevention efforts, you have to look along these lines: peer influence, gender homophily. It's usually a group of 100 to 200 people in a cluster. Mutual friends have twice the influence on each other than isolated friends, and more ties with other people make you more susceptible to the spread of norms or behaviors. Now, in my study, what we did for the Growing Right Onto Wellness trial, and actually in all of my behavioral intervention studies, we use these constructs to see if we can more effectively change knowledge, norms, attitudes, and behaviors. And we intentionally will put a group together, and we actually draw out their relationship to each other, and we see how it changes over time. And what's interesting about that is it creates a ripple effect. So this is just an example of a study that we did looking at, again, preschool children. This is different than the Growing Right Onto Wellness study, but we used this data to help to influence all of our behavior interventions from that time forward. That typically when people enroll in a study, especially the work I do, which is community engaged work, I'm looking at people in free living conditions where they are living and growing and working and playing. And that really determines really where most of health is happening, especially when you're a child. So here you can see that they had some sort of connection to each other. The intervention group had a little bit more than the control group, but after our intervention, while the control group did increase their density of the network, the intervention group increased their density by more than threefold. And that affected whether they continued to use parks and recreation for family physical activity. I just briefly wanted to mention this because it reminds me about when we bring groups together and we're looking at about behavior change. We saw that there was a tendency for parents to form new friendship ties with other parents based on their child's BMI Z-score. Their parents were actually not consciously, but they were tending to meet new people if their children looked similar, and not just gender and age, but also body habitus. That was interesting because that allows you to understand changes in norms. So we wanted to understand in the Growing Right Onto Wellness study how differences in social network typologies might be associated with reducing sedentary behavior. And in this case, we wanted to understand how we could reduce sedentary behavior in parents, because we know that parents of young children really influence how their young children are physically active. So what we saw was that the social network dynamics affected sedentary behavior. So those participants who were connectors, and that's where the circles are, during the 12-week intervention had an average reduction of 30 minutes a day of sedentary behavior 9 months later. Actually, the position in the network changed their ability to change their behaviors as well as others in the group. So I'm going to end by really highlighting that this work that I do, and I just gave you a glimpse of it, really is something that is a team sport. And these are just some of my team members, but I had more than 60 team members just for this one study, and we really represented the world and spoke multiple languages in the lab, and we're very engaged in the community. And I just want to leave you with this comment that when we look at obesity and we look at obesity prevention, we have to remember it's complex, but it's not complicated. So I wanted to leave plenty of time so we could have a conversation. I'm going to stop sharing and open it up for questions. Thank you all. I love the thumbs up and the moving applause because it is strange when you present to a quiet room. But what I'm hoping we can do with our remaining time is engage in a conversation, because what I really wanted to do here was I didn't want to teach you everything you could read about in an article. I wanted instead to spark different ways of thinking and different frameworks we can use when asking and answering questions when it comes to things like obesity prevention and diabetes prevention and other things. Venkat.
Hi, Shari. Thanks a lot. I mean, it's a lovely framing of the complex environment. I particularly enjoyed your DNA methylation project, and we've spoken about it. As I was thinking about it, I mean, have you considered looking at DNA methylation in umbilical cord blood? Because I don't. I mean, I'm hypothesizing here that whatever happens is probably happening in the intrauterine environment. And like Solveig's paper showed, most of the antecedents of childhood obesity are set by age 5. Beyond that, it's very difficult to influence the outcome beyond that.
There are many studies out there that look at cord blood,
Yeah.
and we know so much more about methylation and other ways to capture epigenetics when you look at cord blood. What we don't know is how it changes over time, and really trying to get to what's the granularity in which we should be collecting the data, because we don't understand how quickly these things are adjusting and equilibrating. And that would be incredibly helpful for us, because if we saw, for example, that we really have pretty much set points by the age of 2, that would change how we would intervene in an earlier way. I believe 5 is already too late because you could see, I mean, it's true that our kids who were 3 to 5, we could predict who was going to become obese um, just by their inflammatory markers. So, what could have changed it prior to that is a question. So, were they, how high was that at birth? I don't know that. And I can tell you, and I didn't spend much time talking about the outcomes, but we were able to change nutrition and physical activity, nutrition such that those that were in the intervention group consumed 100 kcals less a day 3 years later than the control group. Both the intervention and control group actually exceeded physical activity guidelines and had more than typically 70 minutes of moderate to vigorous physical activity in the day. Just took them 11 hours to get there. And that's why our collection of the data over time was so important. So how would that change the methylation marks?
Yeah.
And so I think that those are the right questions because it really helps to guide when we intervene and how we intervene. Danny,
Hello.
nice.
Thanks for this. What a treat. Um, a couple of questions, thoughts, and then an offer. So the first question is, you had a sort of single, if I'm understanding you right, for that methylation work, you had a single CPG that was highly predictive of later body size, I think was your outcome.
That's right.
Um, um, did you look at a full methylation score, and was that even better at prediction? And the reason I'm asking this question is, all of us in sort of the genetic epi and clinical genetic epi world have this idea that if we just had sequence data on everyone, we could apply that to our clinical decision making in several ways. What we haven't really talked about is whether or not you could have a methylation array on all of your patients at birth, and whether that would help with clinical decision making, like identifying these high risk groups for early interventions in different ways. But usually it's a score that gives that kind of really high ROC, if you will, rather than a single CPG. So that was my first question.
Yeah. Well, in prior work that I did before I started looking at methylation and epigenetics, I was just looking at genetics and I was trying to recapture some of the work that had been done in the genetic risk scores.
Mhm.
And what's good about a genetic risk score is it works in large populations. But when it comes to the single patient, it's not nearly as useful. So we didn't do that in this population. I think having a head-to-head comparison, and we're always sort of, I'm always grappling because I have a master's of science and health services and think like an epidemiologist, that you find certain patterns that seem absolutely true in populations, and then I can't bring it back to that patient who's sitting in my office. So I think these are really important questions to pose.
Yeah, we see, I mean, you see with other kinds of predictions like in utero exposure to smoking, like those kind of methylation signatures or scores are now like incredibly good at individual level prediction. It's wild, right? So I'm sure it's coming for some of these others. So that was my question, and I think it's really exciting to keep doing the kind of work that would allow that to someday be reality. And then
Yeah.
my offer is in our pregnancy cohorts for autism, we have mom blood during pregnancy, baby cord blood at birth, baby early life blood, and placental tissue already with either whole genome bisulfite sequencing or with epic array methylation panels. We must have done anthropometric measures at age 3. So it feels like we're sitting on data to do any kind of replication like that you might need. So we'll talk offline.
I love that. And please, if you're on today's call, raise your hand. This sounds like a fantastic project for our teams to work on together. What an incredible gold mine.
Yeah.
Thank you. I'm just gonna check here. Would be good to look at methylation in umbilical cord blood. Yes, totally agree. Um, And Venkat, I have to tell you, I learn something new from you every day because you are so good at sharing new information and new ways of thinking. So thank you for that. Holly. Yes. Hi.
Hi, can we ask a question from who's
Oh,
swallowing? We're watching in the middle of clinic. Can you hear us?
I can. Thank you for joining.
Yes, this is our medical student and our resident here in adolescent medicine clinic today. And so since we're sitting here in clinic, I want to ask you about clinical implications for some of what you talked about. Because I was sitting here thinking, like, how do you talk to a family about, genetic risk related to circadian rhythm patterns, or how do you like really, do precision medicine in a way that doesn't feel stigmatizing, or how do you talk to a family about who they befriend, right? So I would love your thoughts about that.
Yeah. So I think that when I'm in clinic, what is most important is that I'm not waiting until a child becomes obese because I know that obesity is an invisible phenotype until it isn't anymore. It's very likely that many of the patients that we see are in a pro-inflammatory state, and that already is setting them on a poorer trajectory away from health and towards disease. So I start with that very first visit, and I really teach families about paying attention to satiety cues. And we know that if you double your birth weight in that first 6 months of life, I'm not talking about premature babies, although that's a separate issue and a lot of challenges there because we often overfeed them to try to get them on the chart, and that affects their adipose tissues. But um, if you grow too quickly in that first 6 months of life, you have a 3 to 5 fold likelihood of becoming obese as a toddler. So I start with that first visit about paying attention to cues. How do you know when to stop feeding your baby? And the number one problem in today's world is that parents are on their screens while they're feeding their babies. So they're not paying attention to those cues. And that builds a different hypothalamus. That changes your satiety set point. So you start there and you pay attention to growth, but I don't talk about growth in terms of a number for weight because I don't want families to fixate on that. I want to fixate on healthy growth and behaviors to support healthy feeding. The then the next thing that I do is when we introduce solids, I give families a sample menu. How much should you eat? It's confusing. How much of a solid? Am I supposed to give 2 teaspoons or the whole jar? And what if they don't drink their milk? Now I'm worried. So I give them examples, and it's very much of a we're in partnership learning together. Uh, and then I really talk about early physical activities of family because children who have parents that are physically active, I don't mean off on their own being physically active, but physically active with their children. Their children are 6 to 9 times more likely to be physically active, not only in childhood, but into adulthood. So establishing family behaviors, and when I say family, writ large, people, even one person that you can rely on. It could be a sister, could be a neighbor, but making sure that there is movement in the body, that's critical as well. And then once I do diagnose obesity, I I have found, and this is different in different languages. Many of the families that I've worked with speak Spanish. And if I say obesidad, that's not so meaningful. But if I say your child's weight is growing faster than their height, and that's not good for their health, may I talk to you about that? What have you already tried? How can I help you? That sort of therapeutic alignment rather than punitive labeling.
Thank you.
I love that you joined from clinic. Thank you, Holly and students. Other questions? Well, I
Shari,
would,
hi,
yeah,
hi, this is Mo. Can you hear me okay?
yeah.
Awesome. This was spectacular. And this is a loaded question, so apologies, but I'm just curious where you land on, as you think about the complexity that you described, the Maha movement wants us all to believe that there's a significant component of that risk that comes from sort of what we are eating and the environmental aspects of what we're eating, whether it be pesticides or other chemicals. And so I wondered whether you had sort of a gestalt feeling about that, based on the data you've seen, and what might be the opportunities to study some of those risks that have been talked about.
And Mo, before you go back on mute,
Yeah,
can you clarify a little bit more? Is your question about the soil in which the food is growing, the processing that we do with food and how that affects things? Is it around that sort of the environment of what we do to take food, real food, and turn it into what we end up eating?
that's my interpretation of it. Yes, all of those.
Okay. Okay, got it. All right. So we have done a lot of work. So I've my colleagues Bill Hierman and I have continued to work on different groups of children and specifically looking at ultra processed foods and using the NOVA classification about what foods we're eating. So if you say someone's eating turkey, but it's actually pressed turkey that has a lot of sulfites, that's actually a different chemical type of type that you're putting into your body versus a turkey that's been minimally processed with maybe no sugar or you have to have salt for preservation. But so likewise, when you eat a carrot, if it grows in a nutrient poor soil, the type of nutrients it has, even though I'm still calling it a carrot, will look different. So I don't know if I'm answering your question, but I do think when we talk about nutrition, we need to be a lot more specific with our language and our understanding. I was thinking about this that I have friends who've got sourdough starter. I don't know if any of you have sourdough starter, but the thing is, you are fermenting that yeast for a much longer time, and that changes the gluten in the bread. So it's still called bread, but the actual biochemistry, the chemistry of the bread is different. So that's true with nutrition, and I think we need to be more specific. And then of course we need to look at ultra processed foods, and the question, of course, is how do you make fresh foods more affordable? But then people have to want to eat them. And I'll tell you, I have kids in clinic that would much prefer some hot Cheetos. They're not, I could give them an apple, but they're not going to eat it. So it's beyond just provision of the food. We need to change palates, which is a totally different thing. And then lastly, I just wanted to mention two studies. Actually, I've got three studies I'm working on. One of them, I'm very fortunate. I work with the Nutrition Precision Health study. It's the first ancillary study to the All of Us study. And it's for 18 and older. And we are collecting everything you can imagine. It is the most exciting, in-depth, detailed understanding of nutrition and intra-individual variation in nutrition. So we take all comers. The first module has 8,000 participants, and we've just finished enrolling our 5,000th participant. And what we want to understand is what's going on with your microbiome, what's going on with your metabolome, your transcriptome, what's going on with what that looks like in your hair and your saliva and your nails and your blood and your feces and your urine. Absolutely everything you can think of. And then we look at area under the curve when it comes to insulin, glucose, IL-6, triglycerides, etc. Then in module 2, we take a subsample of those, and that will be up to 1,200 participants, and we're at about 700 right now. And they get a delivery of 3 different types of meals for 2 weeks at a time, and then a 2-week washout period. And we see how they respond to that. And everything's isocaloric, but that's in free living conditions. And then the third part will take 150 of those that participated in module 1, and we have about 100 right now. And now they're domiciled. So we're actually able to look at the metabolic chamber and energy expenditures, as well as everything else that I just said. So that's going to give us a lot more clarity, I think, but it won't answer all the questions that Mo is asking. And I think we need to continue to ask those questions. The second study that I'm doing is I took this deeply phenotyped cohort of the GROW trial, and I can tell you, every 15 seconds muscle movement for children and and their parents, and we look over time to see how that changed. And I'm using this goes back to my last point that I wanted to make, and that is how do you analyze something when you apply a system science lens? So I'm working with the Brookings Institution, and we're using agent-based modeling. And that allows us to take empiric data, inform a computational model, and then conduct thousands of in silico experiments that can change delayed effects, that can change how different components are working together in a synergistic way beyond interaction. And we're just in our last year of that study. So let me just take one last question. Jean, you're on mute.
Thanks, Shari. This is a very interesting presentation. I appreciate it. I just wondered if you could comment maybe with the last minute, just about how you think that the role of GLP-1s and the new medications and what that's going to do for how we look at pediatric obesity and how we're going to intervene.
Yeah, great. So, I think that it's pretty amazing for people that are severely obese, where nothing else has worked. We certainly have seen some remarkable outcomes when you look at weight. You then have to make sure that we're looking at the whole health and constellation, because of course the concern is that you're losing muscle. And if you're creating and inducing sarcopenia when you're younger, that affects your brain, your bone, and your heart. So I think my concern is that we're focusing only on weight. And it's just about how we utilize it as a tool and understand how the whole thing fits together. I do think, though, Jean, I saw a patient who was 9 years old, who was severely obese and had had such sustained intracranial hypertension when I saw him that he was going blind. And I couldn't get the state to allow him to have bariatric surgery. So that just made me ask the question. I mean, I called and I tried. I really tried because we needed to save his vision. He needed to lose weight in a controlled way, but that was really time sensitive. If I had had a GLP-1, then I would have absolutely used it for that kid, but I wouldn't have used it for the rest of his life. So thank you for the question. Thank you all very much for your attention. I really look forward to actually being in a room all together. And I thank you for the invitation.
Thank you, Dr. Barkin.
Thanks, everybody.